kind of one more general question before we get into the clinical application and disease focus. So we know exosomes and extracellular vesicles have generated growing excitement across the whole biopharma landscape, especially for their potential to deliver therapeutic payloads with precision and minimal immunogenicity. So Aegle has been at the forefront of translating this promise into clinical application. And so from your perspective, what key scientific or technological advancements have helped propel the field from bench to bedside? And what do you see in your experience as the most critical enablers of this progress?
Yeah, so I'd say probably the place where we've made progress is also the place where we need a lot more, and that is in manufacturing. That is something that is challenging. So, if you're trying to develop a stem cell product, for example, you can do that with donors, and there are plenty of donors to supply that need. But then when you're taking that several steps removed and purifying an entity that comes from those cells, the scale up of that can be challenging. And so we've been working with some experts around the world, really, and as have others that are in the field. There are over 70 companies around the world that are working in EVs and or exosomes specifically, which is a subset of EVs. And in trying to develop, ways to leverage things like bioreactors, tangential flow filtration, other more specialized ways of optimizing yield. You have to worry about things being sticky. You have to worry about, yield loss and filtration. So really trying to overcome some of those. And there have been some great advancements in that field, I would have to say, or in that area. I have to say it's still not good enough. We're still practicing in that area, and we need to get better at that. But I think also just there are more and more EV groups or exosome groups that are moving toward the clinic. So we're one of the few that are in the clinic now. There are a few others, but there's, like I said, there are over 70 companies that are working in this area, and a fair number of them are getting ready to launch their first clinical trial. So I think that's going to really start to open up from the regulatory eyes to this. And our experience has been, and perhaps this is a later question, that the regulatory authorities, at least in the United States, which is all we've approached at this point, we have not gone international with this product, have been, because they're so comfortable with cell therapies. Cell therapies have been around, autologous and allogeneic, for 30 years, right? And so, in fact, I had the pleasure of working on one of the very first to ever get approved for many years in the United States, which was Carticel. And there have been many since that time that have come on board. And so they're very comfortable with, how to do that part. It's the next part that gets a little bit more sticky in terms of, well, what do you do with release assays, and with, how do you determine potency and mechanism and define those things? That can be a little bit harder, but I think the breakthrough, if you will, to your original question, is that more and more companies are getting, I guess the agencies are getting more and more exposed to companies, both in the US and predominantly in Europe. There are some companies in Asia, but a lot of this work is going on in the US and Europe. They're getting more exposure to this, and that helps to pave the way, for others to come through and make it an easier way to get approval.
Before we do move on to that clinical application and disease focus part of our discussion, I do want to just briefly draw attention to the poll results, which the question being, we asked our audience, what disease area do you believe holds the most near-term promise for clinical success of exosome-based therapies? And 45.5% are saying autoimmune or inflammatory systemic conditions. Too early to tell is next up, with cardiovascular and rare inflammatory or dermatologic diseases tied at about not quite 14. So do you see that as being kind of representative of the space right now, or are you also in kind of also the too early to tell, or of course dermatologic diseases?
From what I know in terms of what other people are doing, I think one of the main, so I guess first of all, when I look at this list, I see some of the indications that are very amenable to local delivery as opposed to systemic delivery. And I would have to say, at least first out of the gate, that's going to be a lot lower hurdle, both regulatory wise and also just logistically and delivery wise. That's going to be a lot easier. So I think that in the, in the near term promise, the practicalities are that things that can be locally delivered to, say, the eye, the central nervous system, the joint, the wound, those sorts of things are going to probably have a quicker path because it's just an easier path, and then the rest will probably follow. But the second thing I noticed, and has been very exciting to me because I'm a kind of a closet neurodegenerative disorder interest person, so I have quite a bit of interest in that area, and I've been trying to get more involved with it. And some of the things that are becoming very apparent is that when you take, so these EVs take on the cargo, this will sound obvious when I say it, they take on the cargo of the cells from which they come, right? And so if you want to cross the blood-brain barrier and you use specific neuronal cells that have the correct receptors for crossing that locked barrier, those EVs or exosomes will cross the blood-brain barrier. And so that gives them a dramatic advantage for targeting CNS things, results of acute stroke, things like Parkinson's, for example, or maybe even Alzheimer's, although I think that's further away. So I think that's probably coming a second near-term hit because there's a way to target things that we don't have with some other entities. I think further away, and yet the biggest interest, autoimmune or inflammatory systemic conditions, not because there's any lack of potential, but just harder to scale up, as I said, to be adequate for repeated systemic delivery. We don't know about dosing yet, right? So we don't know how easy or hard that will be. I can share that we do have some preliminary data in graft versus host disease in mice, little bitty mice, that is excellent and very promising. So I think there is definitely some potential there. And again, I think the devil's in the details of how do you get that targeted, where do they go when you give them systemically, when you do LNPs or RNA type delivery go to the liver? Does it when you put in EVs? And one of the unique features of EVs versus some other modalities is that, and even the cells themselves, and what I think makes them probably a better choice than just the cells, is that they very quickly, they're little, lipid blebs, right? And they very quickly fuse with the local cells where you put them. So if you put them in a liver, they'll fuse with the liver cells. If you put them in the brain, they'll fuse with the brain cells. And the wounds, they fuse with the fibroblasts, and they dump their cargo into those cells. So what that means is they're one and done. You don't have to, you don't have to worry about residence time. You don't have to worry so much about them being washed away, say in a wound. And when we treat the epidermolysis bullosa, I'll just call it EV now because that's much easier to say. When we treat the EV patients on their wounds, they basically undergo, there's a wrap, a permeable bandage is put over it. It's put in with a pipette tip, so there's no injection of the surface because it's better in these patients who have very fragile skin not to break through and disrupt the wound bed if you can help it. It's put on and just wrapped over, and then the next day they take the wrap off and it's business as usual. So there's not really a huge concern that they're going to take a bath and rinse the product out. So I think that's something that is really helpful for local type applications, but for systemic, it gets a little harder in my mind to figure out where does that go and what does it do. So I think there's just a lot more work there.